transgenic animals and transgenic plants. Genetic manipulation of animal cells. Transgenesis and transgenic animal. Transgenesis may be defined as the introduction of exogenous foreign DNA into the genome, such that it is stable, maintained in a heritable manner. The animals that have been permanently engineered by gene insertion are called transgenic animals, and any foreign genes that are added are called transgenes. Genetically modified, genetically engineered, and transgenic organisms are often used interchangeable, yet they do not mean the same thing. A genetically modified organism is one that had its genetic material altered through any method, including conventional breeding. A genetically engineered organism is one that had been genetically modified using the recombinant DNA technology. A transgenic organisms had been genetically engineered using a foreign gene, usually belonging to a different species. Production of transgenic animals. The first transgenic animals was produced in 1981 by Ralph Brinster and Richard Palmiter. They succeeded in introducing a gene for rat growth hormone GH into the fertilized egg of mice. The injected DNA was constructed to contain the coding portion of the rat GH gene in a position just downstream from the promoter region of the mouse metallothionein gene. A variety of methods have been developed to produce the transgenic animals. All are based on the introduction of the DNA into the single cell that contribute to the development of animal. In mammals, many transgenic experiments have been performed using mice. The method for producing the transgenic mice involves the removal of a fertilized egg or early embryos from donor mother, in vitro transfer of transgene, and then their return to foster mother, where development continues. For most transgenesis, the DNA can be introduced into the mice by 1. Pronuclear microinjection of DNA into the male pronucleus of one cell embryos. 2. Introduction of genetically engineered embryonic stem cell into an early stage developing embryo. 3. Introduction of a retroviral vector in the cell of an early stage embryo. Pronuclear microinjection. In pronuclear microinjection, the transgene is injected into the male pronucleus just after fertilization. The male pronucleus is relatively larger than the female nucleus and closer to the oocyte surface. The microinjection is done using the microneedle. The microinjected transgene randomly integrates into the chromosomal DNA, usually at a single site. Microinjection is done using the microneedle. The microinjected transgene randomly integrated into the chromosomal DNA, usually at a single site and usually at a multiple copies. It is more common for the DNA to integrate after 1 or 2 cell divisions. In such cases, the resulting mouse is a mosaic containing both transfected and non-transfected cells. If the DNA integrates prior to the first division of the zygote, every cell in the resulting animal will contains the same transgene. The surviving intact eggs after microinjections are implanted into a host female mouse. The 3 weeks after the first birth of pups, the genomic DNA from the mouse are analyzed. The mice that score positive by test are then bred to establish for founder transgenic lines. If the DNA integrates after the first division of the zygote, only some of the cells in the egg will incorporate the transgene. However, if those cells contributes to the germline of the embryo, the transgenic gametes will be produced and the subsequent generation of the animal will be transgenic. The figure shows the construction of transgenic mice by pronuclear microinjections. A holding pipette which can hold fertilized oocyte in place, while the microinjection pipette is used to pierce the oocyte, and then the male pronucleus and aqueous solution of the desired DNA is then pipetted directly into the pronucleus. The introduced DNA clones can integrate into the chromosomal DNA. Surviving oocytes are re-implanted into the oviduct of a pseudo pregnant foster female, which have been mated with a vasectomized male. The mating act can initiate the psychological changes in the female, which stimulates the development of the implanted embryo. Transfection of ES cells. The transgenic mice can be produced by the transfection of embryonic stem cells ES. The ES cells are derived from the inner cell mass of the mouse blastocytes and thus have the potential to contribute to all tissue of the developing embryo. The embryonic stem cells can be cultured in vitro and are easy to transfect. The embryonic stem cells remains pluripotent and contribute extensively to all the tissue of the mouse, including the germline, which injects back into the host blastocyte and re-implant in a pseudo-pregnant foster mother. The developing embryo is a chimera that is contains two populations of cell derived from different zygote, those of the blastocyte and the implant embryonic stem cells. This differs from a mosaic in which the cells may be genetically different but are derived from the same zygote. If the blastocyte and the embryonic stem cells are derived from the mice with a different coat color, the chimeric offspring can easily be identified by their patchwork coats. The isolation of blastocyte from the mouse to form a removal of inner mass cell of blastocytes and a culture of embryonic stem cells to then gene targeting or insertion of transgenes into embryonic stem cells, then injection of injection of genetically modified embryonic stem cells into the cavity of blastocytes from a different mouse strain, then the re-implant into the foster mother. Retroviral vector method. The retroviral vector are used as an effective means of integrating the transgene into the genome of a recipient cells. However, this vector can transfer only a small piece of 8 kb kilobase of DNA. The gene knockout. A gene knockout or simply knockouts is a genetically engineered organisms that carry out one or more gene in its chromosomes that has been made inoperative. The primary use of this technique have been to replace a normal gene with a mutant allele or a disrupted form of the gene, thus knocking out the gene's function. There is a difference between the transgenic mice and knocking out mice. In production of transgenic mice, a functional transgene is added to the genome, whereas in the production of knockout mice, a non-functional form of mice gene replaces the functional gene so that the animal does not express the encoded product. A knockout mice differs from a knock-in mice also. Initially, the knockout mice was produced by replacing or disrupting the coding region of the gene, since the mice could be used to study only the effect of the loss of gene, not a specific mutation. In a knock-in mice, a functional gene has been inserted at a selected locus, that is a targeted insertion. The knock-in mice can be transgenic if a new gene is from other species. Process of development of knockout mice. Production of a gene targeted knockout mice involves the following step. 1. Isolation and culturing of embryonic stem cells from the inner cell mass of mouse blastocyte. 2. Introduction of mutant or disrupted gene into the cultured embryonic cells and selection of homologous recombinant cells in which the gene of interest has been knocked out, that is replaced by a non-functional form of genes. 3. Injection of homologous recombinant embryonic stem cells into a recipient mouse blastocyte and surgical implantation of blastocyte into a pseudo-pregnant mouse. 4th, the mating of a chimeric offspring heterozygous for a disrupted gene to produce a homozygous knockout mice. Formation and selection of recombinant embryonic stem cells. A common approach to inactivate a selected gene is to insert an antibiotic resistance gene into the clonal gene of interest. This both inactivates the gene of interest and allows identification of embryonic stem cells that have been successfully transfected. In the engineered insertion construct, the target gene is disrupted with the neo resistant gene, which confers the neomycin resistant. The insertion construct also contains thymidine kinase gene from herpes simplex virus TKHSV, which confers the sensitivity to ganciclovir, a cytotoxic nucleotide analog. The herpes simplex virus TKHSV gene is located outside the target gene. The construct is transfected into the cultured embryonic stem cells. After transfection, the three types of events occurs. One event is homologous recombination. That is, when the DNA vector finds the correct gene inside the embryonic stem cells and integrates into the gene and replaces it. When the homologous recombination occurs, only the target gene disrupts with the neomycin resistant genes will be inserted into the chromosomal DNA of embryonic stem cells. It loses the herpes simplex virus TKHSV gene located outside the target gene. In this situation, the vector has homologous recombinant with the endogenous gene. The second type of event is when the insertion construct goes into an embryonic stem cell and does not find the correct gene but randomly integrates into any part of the chromosomes. This is termed non-homologous recombination. The most constructs will insert at a random by non-homologous recombination rather than by gene transfected insertion through the homologous recombination. In this situation, all three genes will be inserted. The third type of event is when the gene does not take up any DNA at all, so the chromosome are left completely unmodified. The selection of recombinant embryonic stem cells uses the application of neomycin, which kills any non-recombinant embryonic stem cells because they lack the neomycin resistant genes. The herpes simplex virus TKHSV gene helps in selection of homologous recombination embryonic stem cells. The selection with the ganciclovir kills the non-homologous recombinants carrying the herpes simplex virus TKHSV gene, which confers the sensitivity to the ganciclovir. Only the homologous embryonic stem cell recombinant will survive this selection scheme. The figure, the formation of mouse recombinant embryonic stem cells in which a target gene is disrupted. The target gene is disrupted with the neomycin resistant gene. The herpes simplex virus TKHSV gene is located outside the target gene. The construction is transfected into the cultured embryonic stem cells. If the homologous recombinant occurs, only the target gene and the neomycin resistant gene will be inserted into the target DNA of the embryonic stem cells. If the non-homologous recombinant occurs, all three genes will be inserted. Nuclear transfer technology and animal cloning. The nuclear transfer can be used to produce genetically modified organisms. The nuclear transfer involves the replacement of an oocyte nucleus with the nucleus of a somatic cell, which is then reprogrammed by the oocyte and becomes able to recapitulate the whole of development despite the differentiate state of the donor cell. This technique has been used to generate the cloned mammals. The nuclear transfer was first reported in Xenopus in 1952. Since then, it has been widely used in amphibians, primarily for experiments addressing early development. In 1977, a mouse was generated by nuclear transfer using a cell from a very early embryo. In the 1980s, the cloned cell and the cattle were generated using the early embryo cells as donor. The breakthrough came in 1995 when Ian Wilmot and Keith Campbell produces two live lamb Megan and Morgan by nuclear transfer of embryonic cell growth in culture. Within a year, a lamb had been produced from an adult cell. This lamb was called Dolly. Since then, a few lamb and calves have been generated using this technology, some of which are transgenic. The embryo reconstructed by nuclear transfer involves the transfer of single nucleus to an unfertilized oocyte or one cell zygote from which the genetic material have been removed. From livestock, the recipient cell of choice is usually an unfertilized oocyte that has been arrested at a metaphase of the second mitotic division. The first step is to enucleate the recipient oocyte. The genetic material from the donor cell is then introduced into the enucleated oocyte. The next phase involves the activation of the reconstruction embryo. In nature, this is achieved by fertilization and involves calcium mobilization. The activation can be mimicked by a variety of chemical agents such as a bol ester. In practice, this step is often simplified since the electric pulse used to fuse the cells can also activate the embryo. Finally, the embryo is cultured to a stage at which it can be transferred to synchronized recipient animals for the development. Animal cloning. The animal cloning is the process by which an entire organism is reproduced from a single cell taken from the parent organ, and it is genetically identical manner. This means the cloned animal is an exact duplicate in every way of its parent. It has the same exact DNA. This technique used to clone whole animals such as sheep is referred to as a reproducing clone. In reproductive cloning, the research removes a mature somatic cell such as skin cells or an under cells from an animal that they wish to copy. They then transfers the DNA of the donor animal's somatic cell into an egg cell or oocyte that has had its own DNA containing the nucleus removed. The egg can develop into an early stage embryo in the test tube and then is implanted into the womb of adult female animal. Ultimately, the adult female gives birth to an animal that has the same genetic makeup as the animal that donates the somatic cell. This young animal is referred to as a clone. The reproductive cloning may require the use of a surrogate mother to allow development of the cloned embryo, as was the case of the most famous cloned organs, Dolly the sheep. Cloning of sheep. In 1977, Ian Wilmot announced that a sheep had been cloned from a somatic cell nucleus from an adult female sheep. This was the first time that an adult vertebrate had been successfully cloned from another adult. He took cells from the mammalian gland of an adult 6-year-old pregnant Eve and put them into the culture. The culture medium was formulated to keep the nuclei in the cell at the resting stage of the cell cycle G0. They then obtain the oocytes, the maturing egg cells from a different strain of the sheep, and remove the nuclei. The fusion of the donor cell and the enucleated oocyte was accomplished by bringing the two cells together and sending the electrical pulse through them. The electrical pulse destabilizes the cell membrane, allowing the cell to fuse together. The resulting embryo have eventually transferred into the uteri of a pregnant sheep. Of the 434 sheep oocyte originally used in this experiment, only one survived, Dolly. This is appeared that the nuclei of adult somatic cell can be totipotent. The figure. The cloning cell from the nuclear transfer. The nucleus of the ovum is removed with the pipette. The cells from the mammary epithelium of an adult are grown in culture, and the G0 state is induced by inhibiting cell growth. A G0 cell and an enucleated ovum are fused, and the enucleated ovum is grown in culture or in ligated oviduct until an early embryonic stage before it is implanted into the foster mother, where the development proceeds in them. Transgenic plants. The transgenic plants are those plants which carry additional stably integrated and expressed foreign genes from the trans species. The whole process of a transgenic plants development involving introduction, integration, and expression of foreign genes in the host is called transgenic transformation. The combined use of recombinant DNA technology, gene transfer method, and tissue culture techniques has led to the efficient transformation and the production of transgenic in a wide variety of crop plants. Unlike conventional breeding, only the cloned genes of agronomic importance is being introduced without the transfer of other undesirable genes from the donor. The recipient genotype is least disturbed and there is no need for repeated back crosses. This will serve as an effective means of removing certain specific defects of otherwise well adapted cultivars. General procedure used to make a transgenic plants. Agrobacterium mediated transformation. The first step in the process of development of transgenic plants involves the formation of the recombinant plasmid and its transfer to the plant cells. For the recombinant formation, the tDNA needs to be disarmed. To do this, the genes encoded the protein to produce auxins and cytokinins are simply removed from the tDNA fragments. The new DNA can then be inserted between the left and right border respectively. Repeats. The TA plasmid is a large plasmid. The large DNA molecules are difficult to manipulate in vitro, and there is a problem of unique restriction site. However, this problem has been resolved by using two novel strategies: binary vector strategies. The TA DNA does not need to be physically associated with the virulence genes in order to become integrated into the plant genome. In binary vector strategy, the two plasmids are used and both complement each other in the same bacterial cells. The tDNA carried by a small plasmid is transferred to the plant chromosomal DNA by protein coded by the virulent gene carried by another plasmid. Figure the two plasmid system, with the tDNA on relatively small plasmid B and the rest of the plasmid A in normal form. The tDNA plasmid is small enough to have a unique restriction site and to be manipulated using the standard techniques. The plasmid A and B complement each other when present together in the same A. Agrobacterium tumefaciens cells. The tDNA carried by the plasmid B is transformed to the plant chromosomal DNA by protein coded by virulent gene carried by the plasmid A. The co-integration vector strategy. The insertion of a new DNA into a TI plasmid results from the recombination of a small vector plasmid PBR322 and TA plasmid. In this case, the small portion of DNA is used that remains integrated in different plasmid PBR322 or a similar E. coli vector. The recombination takes place through the homologous region present in both plasmid. The figure, the co-integration vector strategy. The insertion of new DNA into a TA plasmid results from the recombination of the small vector plasmid. For example, an E. coli vector and TA plasmid harbored in Agrobacterium. The recombination takes place through the homologous region present in both plasmid. The recombinant tDNA plasmid vector contains a few functional units in addition to the right border and left border elements. These includes 1. A broad host range replication origin such as the R6K. 2. An antibiotic resistant gene for plasmid selection in bacteria. 3. A multiple clone site located between right and left border elements for insertion of a cloned gene. 4. The dominant selectable screening able marker gene with a plant transcriptional promoters for selection of transformed plant cells. A selectable marker gene encoded a product that allows the transformed cell to survive and grow under the condition that kills or restricts the growth of non-transformed cells. Most such genes used in plants are dominant selectable markers that confer resistance to antibiotic or herbicides. A screenable marker gene, also called a visible marker, scoreable marker, or reporter gene, generates a product that can be detected using a simple and often quantitative assay. Such markers are used for a variety of purposes. Example: confirming transformation, determining transformation efficacy, and monitoring gene or protein activity. The selection of cells have transgenes are achieved by two ways. One way is to integrate the selectable markers, a gene which enables the transformed cell to survive under the condition where the untransformed cells would not, that is, negative selection of untransformed cells. Or other way is to integrate the screenable markers, a gene that gives transformed cells an additional capacity which the other cells may not have. For example, the synthesis of some genes which gives some color reactions. The commonly used selectable marker gene includes those conferring the resistance to the antibiotic kanamycin, the NPT encoded neomycin phosphotransferase, and hygromycin HPT encoding hygromycin phosphotransferase, and herbicide glycophosphate EPSP synthetase encoding 5-enol pyruvyl shikimate 3 phosphosynthetase phosphenotricin BA, or codes for phosphenotricin acetyltransferase. Examples of commonly used suitable screening markers: selective agents, streptomycin, streptinomycin gene SPT, hygromycin gene HPT, glufosinate ammonium gene PAT, glyphosate gene EPSP synthase, bromoxynil gene BXN. The agrobacterium that are carried a recombinant plasmid with both a selectable marker and a desirable transgenes are incubated in a culture with plant cells. The wounded plant cells at the edge releases substances that attracts the agrobacterium and causes them to inject the DNA into these cells. Only those plant cells that takes up the appropriate DNA and expresses the selectable marker survives to proliferate and forms a callus. The growth factors are supplied to the callus induces it to form shoots and roots and grow into adult plants carrying the genes. The figure: an agrobacterium plasmid that normally carries the tDNA sequence is modified by substituting a selectable marker such as a kanamycin resistant gene and a desirable transgene between the 25 nucleotide pair tDNA repeats. When the agrobacterium recognizes the plant cell, it efficiently passes a DNA strand that carries these sequence into the plant cells using the special machinery that normally transfers the plasmid tDNA sequences. Only those plant cells that takes up the appropriate DNA and expresses the selectable marker gene survives to proliferate and forms a callus. The manipulation of a growth factor supplied to a callus induces it to form a shoots that subsequently root and growth into adult plants carrying the transgene. Application of transgenic plants. 1. Development of insect, virus, and herbicide resistant plants. Insecticidal resistance. Through genetic engineering, it is possible to develop crops that are intrinsically resistant to insects. Several different strategies has been used to confer resistance against an insect predator. One approach involves a gene for an insecticidal protoxin produced by one of several subspecies of bacterium Bacillus thuringiensis. The crop plant has been engineered to express the insecticidal toxin gene of Bacillus thuringiensis so that the insecticide attempting to eat these plants are killed. Bacillus thuringiensis during this sporulation forms intercellular crystalline bodies that contains an insecticidal proteins called delta endotoxin. The delta endotoxin protein Cry1, Cry2, Cry3 and others accumulates in the bacterium as an inactive precursor. After ingestion by the insect, this protoxin is cleaved by proteases, resulting in short version of protein that displays the toxic activity by binding to the side of insect gut and damage the surface epithelium. Virus resistant plants. Several approaches has been used to engineer plants for virus resistance, such as introduction of coat protein genes, antisense RNA approach, ribosome mediated protections. Of these strategies, the use of coat protein genes has been the most successful. The transgenic plants having the virus coat protein gene linked to a strong promoter have been produced in many crop plants such as tobacco, tomato, alfalfa, potato, etc. The first transgenic plants of this was tobacco, produced in 1986. It contains the coat protein gene of tobacco mosaic virus. When these plants are inoculated with a tobacco mosaic virus, the symptoms either fail to develop or were considerably delayed. The effectiveness of the coat protein CP gene is a confirmed virus resistance and can be affected by both the amount of coat protein produced in the transgenic plants and by the concentration of virus inoculum. Most likely, the resistance generated by the coat protein is due to the blocking of process of uncoating of virus particles, which is necessary for the viral genome replicated as well as expression. However, other effects seems to be involved in producing the coat protein mediated virus resistance. One such mechanism appears to be the prevention or delay of systemic spread of viruses. In other approach, the transgenic expression of a dysfunctional viral movement protein MP is used to make the plant virus resistant. The resistance confers by the transgenic expression of a dysfunctional movement protein is likely due to competition for plasmodesmata binding site between the mutant MP mutant movement protein and wild type mutant movement protein of the inoculated viruses. An interesting attribute of the movement protein mediated protection is the broad spectrum efficacy of the resistant mechanism. The protection conferring by the mutant movement protein of tobacco mosaic virus, for example, mediates resistance to other viruses also. Herbicide resistance. Many crops have been engineered for resistance to herbicides such as glyphosate. The glyphosate trading name group up is a non-selective herbicide that inhibits the 5-enol pyruvyl shikimate 3-phosphate EPSP synthetase, a key enzyme in the biosynthesis of aromatic amino acids in plant. The 5-enol pyruvyl shikimate 3-phosphate synthetase converts the shikimate and phosphoenol pyruvate into 5-enol pyruvyl shikimate 3-phosphate, a precursor for synthesis of aromatic amino acids tryptophan, tyrosine and phenylalanine. The glyphosate competes with the phosphoenol pyruvate for binding with the 5-enol pyruvyl shikimate 3-phosphate synthetase. Two approaches have been used to engineer resistance so that the herbicide can be used for weeding control without damaging the crops. In the first approach, the target protein of the herbicide, the 5-enol pyruvyl shikimate 3-phosphate synthetase, can be overproduced so that the resistance occurs because of having much more enzyme available for the cell. A second approach results from the expression of the mutant version of 5-enol pyruvyl shikimate 3-phosphate synthetase that is resistant to the herbicide within the cell. 2. Development of stress tolerance plants like oxidative stress and salt stress. 3. Modification of plant nutritional content like amino acid, lipids, vitamins, and iron. The use of genetic engineering technique allows scientists to develop the plant with improved nutritional quality. For example, rice is extremely low in vitamin A. Potricus and a berry developed a genetically engineered rice, popularly known as a golden rice, which is enriched in pro-vitamin A by introducing the three genes involved in the biosynthetic pathway for carotenoids, the precursor for vitamin A. To obtain a functional pro-vitamin A beta-carotene biosynthetic pathway in rice endosperm, the gene coding for the phytoin synthetase PSY and lycopene cyclase LYC, both from Narcissus pseudo-Narcissus, together with the gene coding phytoin desaturase from CTRL from bacterial Yervinia iridovora were introduced. This combination covers the requirement for beta-carotene synthesis. Additional experiment later reveals that the presence of LYC was not necessary because PSY and CTRL alone were able to derive the beta-carotene synthesis as well as the formation of further downstream xanthophylls. 4. Plant as a bioreactor for production of antibodies polymers. The plants are easy to grow and can generate considerable biomass. This is the reason that the transgenic plants are used to produce commercial protein and chemicals. The plant have been used to produce monoclonal antibodies, the polymer polyhydroxybutyrate, which can be used to make a biodegradable plastics like materials, and a number of potential therapeutic agents like a human potency anticoagulant, human herodin variant 2 anticoagulant, and a human erythropoietin anemia, the human alpha interferon, hepatitis C and B, and the human growth hormone dwarfism. The term plantibodies is used for antibodies that are synthesized in the transgenic plants. The difference between the plantibodies and a edible vaccine is that the plantibodies are pre-made antibodies that are produced in the transgenic plants, whereas edible vaccine promotes the production of specific antibody by human immune system once the vaccine is administered to the patients. The plantibodies are advantageous for plants who are immunosuppressed and are able to produce antibodies even after they are vaccinated. 5. Synthesis of edible vaccines. The commercial vaccines are expensive to produce and package and require trained personnel to administer injections. So, it would be advantageous if vaccine could be delivered inexpensively on a broad scale in an edible form. Example, as part of a fruit or vegetables. An edible vaccine, in contrast to traditional vaccine, would not require elaborate productions, facilitate purification, sterilization, packaging, or specialized delivery system. The gene encoding of orally active antigenic protein is isolated from the pathogen, and a suitable construct for constitutive or tissue specific expression of the gene is prepared. The gene is introduced and stable integrated into the genome of selected plant species and expressed to produce the antigen. The appropriate plant pot containing the antigen may be fed raw to humans to bring about immunization. A concern with oral vaccine is the generation of a protein component in the stomach due to low pH and gastric enzymes and the gut before they are elicit immune response, but the rigid plant cell wall would provide protection from the intestinal degradation. 6. Delayed ripening. The genetic engineering is used to delay ripening in fruits. A few gene controls the ripening process. One of these, encoding the enzyme polygalacturonase, is involved in slow breakdown of a polygalacturonic acid component of cell wall in the fruit pericarp. Its effect results in a gradual softening that makes the fruit edible. However, longer the enzyme can act on the cell wall, the softer and more overripe fruit will become. Therefore, if the effect of the enzyme can be delayed, then the fruit will ripen more slowly. Tomato have been engineered so that they expresses less of the polygalacturonase enzymes. This was achieved through the antisense technology. Using the antisense technology, Calagin, Fresh Inc. USA has permanently introduced an antisense copy of the gene for polygalacturonase with the aid of Agrobacterium tumefaciens. The expressions of antisense copy of the gene gives antisense RNA molecules. The antisense RNA has the opposite sense to mRNA sense RNA. The presence of complementary sense and antisense RNA molecules is the same cells can lead to the formation of a stable duplex which can interferes RNA processing or possibly translations. The antisense RNA binds to the normal sense RNA, preventing the tomato from making the usual amount of polygalacturonase. This genetically modified tomato markers used under the trade name flavor savor well, therefore resist softening and have extended shelf life. Production of bioplastics. The bacteria alkaligens eutrophus produces a polyhydroxybutyrate PHB, a biodegradable and renewable biopolymer. The gene from alkaligens eutrophus that encodes for an enzyme responsible for the biosynthesis of polyhydroxybutyrate is being transferred to plants from production of biodegradable plastics. Antisense technology. The antisense agents inhibits the expression of a target gene in a specific sequence specific manner. The potential of oligodeoxynucleotide to act as antisense agent that inhibits the viral replication in cell culture was discovered by Zamicnik and Stephenson in 1978. Since then, antisense technology has been developed as a powerful tool for target validation and therapeutic purpose. Three types of anti-mRNA strategies are currently in use. First, the use of single stranded antisense oligonucleotide. The antisense oligonucleotide usually consists of 15 to 20 nucleotide, which are complementary to the target mRNA. Blocks translational of target mRNA in a sequence-specific manner, either by steric blocking of translation or by restriction of the bound mRNA via RNase H enzyme. The most antisense oligonucleotides are designed to activate RNase H, which cleaves the RNA moiety of the DNA RNA heteroduplex and therefore leads to destruction of the target mRNA. In addition, the antisense oligonucleotides that do not induce RNA H cleaves can be used to inhibit the translation by steric blockade of the ribosomes. When the antisense oligonucleotides are targeted to the 5' terminus, binding and assembly of translation machinery can be prevented. Secondly, the triggering of sense strand mRNA cleaved by incorporating the ribosome catalytic center into the antisense RNA. A few ribosomes have been characterized, including the most studied form called the hammerhead ribosome, first isolated from the vibroid RNA. Thirdly, the RNA interfered includes by the small interfering RNA molecules. This naturally occurring phenomenon, a potent sequence specific mechanism for post-translational gene slicing, was first described from the nematode worm Canor habditis elegans. The figure. The comparison of different antisense strategy, antisense oligonucleotide block translation of mRNA or induce its degradation by RNase H, while ribosome possesses a catalytic activity and cleaves the target RNA. The RNA interference approaches are performed with siRNA molecules that are bound by RISC and induces degradation of target mRNA. Transgenic animals and molecular forming. A transgenic animal carried in all its cell a foreign gene that was inserted by the laboratory technique. Each transgenic organism is produced by introducing the cloned gene composed of deoxyribonucleic acid DNA from microbes, animals, or plants into plants and animal cells. The transgenic technology offers methods that allows the transfers of gene between different species. 1. Transgenic mice in immunology. The introduction of rearranged immunoglobulin Ig gene into the transgenic mice had helped enormous to clarify the mechanism of rearrangement of Ig genes and the importance of this phenomena to the regulation of the expression. The study indicates that the expression of rearranged heavy chain gene in T cells indicates that the inability of the T cells to express heavy chain genes is related to the absence of gene rearranged in this lineage. To transgenic mice in oncology, the insertion of oncogenes and proto-oncogenes into viruses of mice, followed by tracking their effects in the normally differentiating cells of an intact organism, has contributed in understanding the neoplastic disease and its relationship to aberrant gene expression. 3. The transgenic mice as animal model of human disease. Animal model for human illness are useful for studying the pathogenesis of disease as well as for developing and testing new therapies. Human disease can be induced in transgenic mice by expression of a transfer gene or by insertional disruption of a endogenous sequence. Some example of model created by the transgene expression are listed below. A. Hepatitis B. Hepatitis B is a human disease that lack a readily workable animal model. Introduction of the hepatitis surface antigen gene into the mice results in the development of transgenic mice that mimics the carrier state with the production of hepatitis B surface antigen in the liver but with the absence of disease. B. Progressive multifocal leukoencephalopathy. Progressive multifocal leukoencephalopathy can be recreated in mice by injection of the IC viral genome. The another disease of the nervous system, neurofibromatous, can be induced in the mice by introduction of the gene for tyrosine aminotransferase TAT of HTLV1. 4. Transgenic animal for xenotransplantation. The use of animal origin for human transplantation is called xenotransplantation. In 1992, a Cambridge biotechnology company, Mutron, produces a genetically engineered pig called Astrid that carries gene from the human immune system. The organs from genetically engineered pig contains genes that produces a human protein. Such organs would not be recognized as a foreign when transplanted into primate or man. 5. Molecular priming. The forming relies on two related technologies: transgenesis and cloning. The transgenesis allows scientists to create animals with an extra foreign gene, which makes the protein of interest incorporated into their own genetic material. This gene is attached to other regulatory genes, so it only produces the foreign protein in specific tissue, usually the mammary gland or the liver. The transgenesis is very imprecise and is successful in around only 4 to 6% of experiment. The remaining 94 to 96% of animals are unwanted and killed as they do not carry the transgene in the current position, or in many cases do not have the transgene at all. Since transgenesis is so unreliable and expensive, the cloning is used to make copies of those animals that have been successfully genetically modified. The proponents of this technology envisage these techniques being used together to create entire flocks or herds of cloned transgenic animals, all producing commercially valuable proteins. However, the precise nature of these technologies and the tremendous loss of the life they entail are providing considerable stumbling blocks. One, bioforming. This is an experimental application of biotechnology in which the organism are genetically modified to produce a pharmaceutical proteins and chemicals they do not produce naturally. A few examples includes a contraceptive, a potent growth hormone, a blood clotting agent, a blood thinner, industrial enzymes, and vaccines. Two, a forming with animals. Some animals have been genetically modified to produce human proteins in their milk. The animals are like a living pharmaceutical factory, and the technique is often referred to as a farming animals, which may be used to include cow, sheep, pigs, goats, rabbits, and mice. The section of human DNA containing the genes for the specific protein requires in the is injected into the host animal embryo. The embryo is then placed into the uterus of the surrogate mother, where it develops to full term. As an adult, the animal produces the human protein in its milk. The animal is milked, and the protein is purified from the milk for pharmaceutical use for humans. The genetically modified animal produced for pharmaceutical production should be able to produce the desired drug at high level without endangering its own health and pass this ability to its offspring. The first use of this technology was in 1987, and with mice that produces a human drug, the therapeutic plasminogen activator TPA, which is used to treat blood clots. Another research has included cows that produce human serum albumin used to maintain fluid balance in the blood, and lactoferrin, which is a protein from breast milk that promotes infant growth. The sheep that produce a few protein including the factor 8 and factor 9, which are essential for blood clotting, the natural anticoagulants used during the heart surgery, the proteins to treat lung and liver disease. And 3, rabbit that produces human interleukin 2, a protein essential in the immune response to infection and may be successful in fighting some type of cancers.